مرحبا بكم يا شباب والصبايا. اسمي الدكتور واديم زايتشيكوف، وأنا أرحب بكم من بيت روسي في بيروت لحضور محاضرة دكتور سيرغي سيديخ تحت عنوان علم وراثة نوعية حياة، which تناظر عالم أحياء جزيئية في إطار عام علوم والتكنولوجيا في روسيا. كما تعلمون لقد بدأنا في لبنان اختيار تنافسي، وفتحنا مجال للتسجيل لطلاب يحبين يدرسون على حساب الحكومة الروسية في الجامعة الروسية. نحن فتحنا مجال للتسجيل على الموقع الإلكتروني educationinrussia.com. عندكم كما يوجد أسبوعين لتسجيل على هذا الموقع، وتستطيعوا تشاركوا في اختيار تنافسي للحصول من الدراسية الروسية لدراسة في الجامعة الروسية في عام دراسي 2022-2023. بما أننا نحب أن نساعدكم في تعرف على الاختصاصات المستقبلية والاختصاصات المختلفة التي يمكنكم اختيارها، نقوم خلال هذه الفترة بإعطاء سلسلة من المحاضرات حول الاختصاصات الجديدة مثيرة للاهتمام. اليوم سنناقش مع باحث في جامعة نوفوسيبيرسك الحكومية، الدكتور في علوم الأحياء سيرغي سيديخ، القضايا المتعلقة بكيفية تأثير معرفة علم وراثة على نوعية حياة. ستتطرق دكتور سيديخ إلى التطورات في علم الوراثة التي بدأت البشرية تستفيد منها، كما سيخبرون لماذا يكفي إجراء اختبارات الجينية مرة واحدة في عمر، وكيف تساعد جينات في محاربة السرطان وعن الطب الشخصي. بدي أعطيكم فكرة بسيطة عن جامعة نوفوسيبيرسك الحكومية. جامعة نوفوسيبيرسك الحكومية تحتل مرتبة 288 بين 20,000 جامعة في تصنيف QS، ومرتبة الثالثة في روسيا. تدخل جامعة نوفوسيبيرسك الحكومية في 24 تعاونا دوليا، 16 منهم في مجال فيزياء الجسيمات وفيزياء الفلكية. تتعاون مختبرات البيولوجية والطب في جامعة نوفوسيبيرسك الحكومية مع مركز حكومي علمي لعلوم فيروسات وتكنولوجيات الحيوية فيكتور. فهذا المركز بحثي هو الذي عمل واكسين ضد كورونا، فأساتذة الدكتوراه من الجامعة ساهموا كثير كمان في هذه الواكسين. ترتبط جامعة نوفوسيبيرسك الحكومية ارتباطا وثيقا بفرع أكاديمية علوم الروسية في سيبيريا، شاركوا طلاب جامعة نوفوسيبيرسك منذ سنواتهم الأولى في بحوث علمية في فروع أكاديمية علوم روسية في سيبيريا، وعندما يدخلون جامعة وأثناء دراساتهم يعملون في علوم وتحتوي مجموعة يعملون في علوم وفي بحوث علمية. وكمان تحتوي مجموعة مكتبات علمية في جامعة نوفوسيبيرسك الحكومية على أكثر من 900,000 كتابا، وهو ما يزيد عن 27 كيلومتر من أرفف كتاب. يعمل خريجو جامعة نوفوسيبيرسك الحكومية في جامعات ومراكز بحث أجنبية الرائدة. تحتل جامعة نوفسibirsk الحكومية مرتبة السادسة بين الجامعات الروسية من حيث رواتب متخصصين الشباب عاملين في صناعة تكنولوجيا المعلومات، ومرتبة ثامنة في اقتصاد ومالية. تحتل نوفسibirsk كمدينة مرتبة 92 في قائمة أفضل مدينة طلابية في العالم وفقا لتصنيف QS Best Student Cities 2019. فالآن أحب أن أرحب دكتور سيرغي سيديخ. Hello, Sergey Sidikh. Welcome you from Lebanon. We are grateful that you found the time to give this lecture for our students today. I am sure it will be very interesting and beneficial for them. Please, Sergey Sidikh, you can start your lecture. Thank you very much.
Thank you for the invitation. I'm very pleased to tell about genetics and quality of life. Can you please let me to demonstrate my screen to show the presentation? I will introduce myself in short. So I'm biologist, I'm molecular biologist, I'm biochemist, and I defended my PhD dissertation field of biochemistry. And now I'm investigating the exosomes from the milk and its protein and RNA content. Включите меня, пожалуйста, пока с презентацией я не могу презентацию показать. So I need.
Okay. Just a minute.
Okay.
Thank you.
So this is my presentation. I want to show you in short the new building of Novosibirsk University. We have a faculty of natural sciences. In Novosibirsk University is the first faculty that was in Novosibirsk University since its organization in the middle of the century. Now there are approximately 12 different faculties. And on Faculty of Natural Sciences, we have two departments, the Department of Chemistry and Department of Biology. Also, we have an Institute of Medicine, where I talk lectures for foreign students about the cell biology. And we have many other faculties and institutes, and you can go through internet and investigate this information for yourself if you're interested in scientific education. So a few words about the genetics. The genetics is the science of heredity and variability. And in the history of genetics, we know that the science was established in the early 20th century, in 1905. The term genetics was invented, and after that, in 1909, the term gene was invented. So the gene is younger than genetics, and the genetics is not the science about genes, it's the science about heredity and variability. And if we look on this picture, we can easily understand what is heredity. Heredity is when the children looks like the parents. And here we see the granddaughter, daughter, and mother. And the grandmother, they look like each other, and we understand they are relatives. And on this picture, we can see the Galapagos finches, the Darwin finches, the birds that were described by Charles Darwin on the Galapagos Islands during his famous journey throughout the world. And you can see how these finches, they have different, they all are relatives, but they have different shapes of the head, and they have different hooks, and they because they are fed on different sources. So these ones eat the insects, this eat the seeds, and the ancestors not look like its parents. So genetics was invented in 20th century, and we know that the ideas of Darwin were made in the 19th century, and also in 19th century, the Mendel's law of heredity were postulated. So maybe you know, maybe you remember from the school program how the Gregor Mendel, he was a monk, the monk from the Czech Republic. He crossed a species of the bees, and he obtained all the flowers or the seeds in the F1 in first generation, and after that, when he crossed them, when he hybridized them, these hybrids of F1, he obtained the 3-to-1 in first generation. In case of the monohybrid crossing, and in case of the dihybrid crossing, he obtained the ratio 9:3 to 3:1, and he obtained different types, different combination of phenotypes. And now we know that different ratios of the genomes. And now we understand that all these characters of the pea plants that Mendel crosses, which is each other, are located in 7 different chromosomes of pea, and there are the only 7 pairs of chromosomes in the pea plant. So this characters are the color of flowers, the color and shape of seeds, the shape and color of pod, the position of flowers, and the length of the stem. The Mendel didn't knew about the amount of chromosomes in the nucleus of pea plants, but now we know that it's this explains the phenomena that Mendel described in the middle of 19th century. So the next big step in genetics is the theory of chromosomal basis of inheritance. It was invented by Thomas Hunt Morgan. He's a Nobel laureate, American famous geneticist. He crossed the Drosophila melanogaster, this fruit fly, that's now the most important and most common object in modern genetics. These flies are very good object because they have very fast generations. They have very simple food that they need for their lives. They have small bodies. You can easily locate them under the microscope. You can investigate the color of eyes, the shape of the body, the shape of the wings, and so on and so on. So on. They have thousands of mutations, and the first mutation that was connected to the sex chromosomes was analyzed as a result of experiments on Drosophila melanogaster. And here you see the flies with the red eyes, and here you see the flies with the white eyes. And when the Thomas Morgan crossed the flies with the white eyes and with the red eyes, he obtained different results in different cases when the females were with the red eyes and when the males were with white eyes. He obtained the, such situation when most of the flies were 3 parts was with the red eyes and just 1 part was with white eyes, like in the Mendel's ratios. But you see here only the males have the white eyes, and both the females and males may have red eyes. So this can be easily explained if we postulate that the gene that encode the characteristic of the color of eyes is located in the X chromosome, in sexual chromosome of Drosophila melanogaster. And in the back rows, and you take the females with the white eyes and males with red eyes, they all offspring will have the red eyes. So it's the chromosomal basis of inheritance, the work of Thomas Hunt Morgan and his colleagues that brought them the Nobel Prize. So the other investigation of the middle of 20th century is the double helix. And here we see 2 researchers, 2 biochemists and physicists, James Watson and Francis Crick. They described the structure of nucleic acid of DNA. It's using the X-ray data of Rosalind Franklin. They published a paper on the 24 October 1953 when they described the structure and mechanism of later the mechanism of replication of DNA. And this is the birthday of DNA, day of DNA. So James Watson published a famous book, The Double Helix, a personal account of the discovery of the structure of DNA. And it was published in the middle of the 20th century, and in 2000s, he published another book, The Genes, Girls, and Gamma, after the double helix. And I recommend to read at least the first book, The Double Helix, to everyone who is interested in the history of molecular biology. So we know that DNA is a double helix. It's have the sugar phosphates from the outside and hydrogen bonds with the nitrogen residues inside the double helix. And we see here that between the adenosine and the thymine, there are 2 hydrogen bonds, and between the guanosine and cytidine, it's 3 hydrogen bonds. And these 2 strands of DNA are connected very tightly due to the amount of hydrogen bonds is very, very, very big. So our genome is approximately 3 billion pairs of nucleotides. The project of the end of the 20th century is the Human Genome Project, and the beginning of the 21st century, the Craig Venter and other researchers start to sequence all the nucleotides in all of human chromosomes. And here you see the human chromosomes under the light microscope. And the first article was published in 2001 about the initial sequencing and analysis of the human genome. And after that, the whole genome sequence was published, and all the genes were predicted. And here you see the shelf in the library where all the human chromosomes are published as the sequence of letters A, T, G, and C. And here you see that the information of human genome is very, very enormously large. So how to understand this information and how this information may give us something good for our health, we will discuss in this lecture. So in 2020, Two researchers in chemistry obtained the Nobel Prize for the development of a method for genome editing. It were the Emmanuel Charpentier and Jennifer Doudna. They are American researchers, and they developed a method how to make the genetic scissors, how to make double threading breaks in the targeted sequences of DNA, and they used the system from the Streptococcus adaptive immune system that keeps its genome, helps to keep it safe against the viruses. And now this technology is very promising, and a lot of papers already is published for the theme of the genome editing and for use it in to resolve some tasks in genetics. So the non-genome sequencing, I think that everyone knows what this means. It's the sequencing is the detection of the nucleotide sequences in DNA or RNA. And you see that the cost of genome sequencing was hundreds of millions 20 years ago, and now it may cost approximately $1,000 for the exome sequencing, and you will obtain all the information about all of your genes. What it made before, and how to make this genetic analysis. So you see here, you need to put some saliva, some cells from the buccal epithelium of your mouse. You need to fill such test tube for maybe 10, 15 milliliters of saliva, or you can use this rod to pick the sample from your mouth. After that, you put it in special solution with the conservants, with the preservatives, and so on. And after that, you isolate the DNA, you wash the DNA in case of the centrifuge filter isolation of DNA. And after that, you elute the DNA, you obtain the solution of your genome DNA. Usually such operations take place in such laboratories with very high scientific level of high level of scientific equipment, and with the specialists in the white lab coats and so on, with automatic dosators and so on and so on. So if you want to become a geneticist, you can imagine you can work in such laboratory. And there are several types of genetic analysis. The most old one is the Sanger sequencing, when you use first the PCR with fluorescent chain genetic D-desoxynucleotide triphosphate. You obtain the short and the long sequences from the small fragment of the gene. You make the separation by capillary electrophoresis. After that, you analyze all the nucleotides in the fragments you have obtained. The other powerful method is using of the DNA microarray on DNA microchips. You hybridize the molecules with different sequences, with different oligonucleotides that are located on the microchip, and after that you can tell which mutation is or the allele of gene on every region of your genome. And sometimes you can use special markers to make the forensic investigations and so on, to analyze different parts of chromosomes, different alleles, and tell whether the child is the daughter of this mother or this father, to compare the relatives with their genetics information. So one of the leading services for genetic analysis is 23andMe, Several years ago, I obtained such kit from the United States. I put some saliva in this test tube and sent it back to the United States. And in several months, I obtained the results of my genetics. And I want to share with you this information and explain how I am a molecular biologist. How can I interpret this data and whether this information is relevant and interesting for me and for maybe other people. So the first one is very interesting. It's the analysis of my ancestry. So there are different locuses, the fragments of our genes that is associated with different ancestry, with different pedigree, with different regions of the different countries and inside some countries. And as I can see, in my case, the most of my genes are European genes, and most of my relatives come from the Tatarstan and from the Ukraine. So it wasn't a surprise for me because my grandmother was, she lived near Ufa in the Republic of Bashkortostan. That's why it's absolutely normal for me to have such relatives. I have 4% of my genes that are Western Asian and North African. I have 2% of genes that are Central and South Asian, and maybe 2% of in East Asian and Indigenous American. And I have some persons of DNA that is unsigned. We don't know this. Fragments of genome are not associated with any ancestry. And here you see the most of my ancestry is from the Siberia, from the Central Asia, from the Italy, and from Eastern Europe. So you can we can see it even from the ears of this diagram. Also, we can I can see how my chromosomes, which of the chromosomes contain which information, which ancestry, and you can see here that the most of the chromosomes have the European genes. The 6th chromosome and the 18th chromosome contains the Western Asian and North African genes, and several parts of 7th, the 12th, and 16th chromosome contain the DNA from Central and South Asia. So, will this help me to live better? I'm not sure. Okay, the other very interesting information from my genetic analysis is the amount of genes that came from Neanderthals. So maybe all of you heard about the Neanderthals. If our ancestors, the human-like monkeys, and our ancestors, and we know that our human ancestors have crossed and have hybridized with the Neanderthals. That's why we have some genes for the Neanderthals. And it was a big surprise for me. I have more Neanderthal DNA. There's 90% of other customers of 23andMe. What does it mean? This means nothing, but you know, that's especially when we know that the Neanderthal ancestry accounts less than 2% of my DNA. So it's very minor fraction, but I have much more than most of the customers of 23andMe. Out of 3,000 variants tested with 23andMe, I have approximately 300 variants, maybe 1/10. So I have a lot of information about the hereditary diseases, and I have not a single variant that may tell in the favor of hereditary diseases in my organisms. And as you see here, such the diseases like the Bloom syndrome, the cystic fibrosis, the Fanconi anemia, the Gaucher disease, the hereditary fructose intolerance, the phenylketonuria related disorders, the sickle cell anemia, and Sjögren-Larsson syndrome. So I have no any single variant of these genes. It's good information, but most of these hereditary diseases start in the early adult age, maybe in 10 or 15 years. So I was approximately 30 years ago when I passed this test, and the most of these diseases cannot be my disease because of the they were not showed each other during my young ages. So I have slightly increased risk of hereditary predisposition in case of age-related macular degeneration and celiac disease, but I don't see any symptoms of these diseases. I'm 35, and I will be variants of hereditary predispositions also and the main ones. It is some variants of BRCA1 and 2, the hereditary amyloidosis, gamma-hepatosis, thrombophilia, the Parkinson disease, and the typical likelihood of the type 2 diabetes. So there are different health and traits, and this service tells that my genetics predict. You see this sentence. Your genetics predict that in 99% you don't have a chance to have red hair. But you can see I don't have red hair, and it's this genetic information. This information is now proven with the genetic information. The high chance that I have very fair skin, or maybe moderately fair skin. I really have not very dark skin. I have the slightly wavy hair texture. I have dark brown hair, maybe light brown hair. I have not very long hair, and I have very few freckles. It's also I knew this information before I passed the genetic test. So usually the mosquitoes bite me not so frequent, but sometimes they do so. I usually don't have the motion sickness, and I don't have sneeze reflex, and this also helps and traits that are diagnosed through my genetics. The other interesting health and traits is that in chance of 74%, I will not have the hair loss or thinning before 40. I will not have bald spot in chance of 84%. I have dry flaky earwax, and it's a mongoloid symptom. I have some mongoloid relatives. I have high chance that I have brown or hazel eyes. I have very little hair on my back. It's true, and genetics predicts the same. And also, I don't have cheek dimples. The chance of 65%, and I can say it with 100% chance. So very interesting information. I can smell the odor of asparagus in my urine. I never try to eat the asparagus. The next time I will definitely try to feel the odor. I can taste some bitters, and I shouldn't prefer the vanilla or chocolate ice cream, but as I say, I prefer the vanilla one, but not so much. And the system says that I should prefer salty, but not sweet. I'm not sure that it's true, but I'm not sure that the opposite is true. Also, very interesting results about the wellness. I'm likely to flush after the alcohol consumption, and this name alcohol flush reaction is also a mongoloid symptom, and I have this symptom after drinking, and I have wrote the article in Wikipedia about this in Russian. That's why it's not very relevant information. I'm deep sleeper. I can sleep deep for a long time. Okay. I'm tolerant to lactose, as says the 23andMe, but I know that I'm not tolerant to lactose, and my daughters also are not tolerant to lactose. I have a muscle composition common in elite power athletes. So this means in case when my position is analyzed to take part in the Olympics, I can join because I have athletes, and sleep movement, the genetics say it's likely over or less movement. So how should I live with all this data? I have a file. It's the text file or the Excel file, 15 megabytes. And for every my single nucleotide polymorphism, the 23andMe tells my genotype. So I can use it in different services. I can upload this information and look on my traits, but I can say nothing more interesting that I can smell the odor of asparagus in my urine. So, but what usually medicine and genetics want to tell us in case of the genetic data? And here I should talk a few words about the personalized medicine. It's a medicine when we investigate the patients that have the same diagnosis and same course of treatment. But these patients, they can have differences in drug metabolism, in genetics, in age, in race, in their weight, in sex, in accompanying disease. And the patients with this diagnosis, with this same course of treatment, may have two variants of working with this treatment. They can have effective treatment with this drug and no side effects is the best result, and all people want to give such treatment without the side effects and which is effective. The worst situation is when you have effective treatment but you have side effects, and you should obtain the other pills that have smaller side effects. Maybe you should need to decrease the consumption of the drug. There are two bad situations when the medicine doesn't cure and you just don't have any side effects, and sometimes the medicine doesn't cure but has side effects. So these situations are very bad, and the current situation that using the personalized medicine, the medicine wants to tell you whether you should use this preparation or not, whether you will have or not the effect of medicine and the any of side effects. So in case of the personalized cancer treatment, first you need to make the molecular profiling of the tumor, and the researchers investigate the prognostic markers, the markers predictive for drug sensitivity and resistance, the markers that are predictive of the adverse effects. And when they generate all this data, every person and every group of persons will obtain personalized cancer treatment. And you see here are the people with the yellow color. They will obtain a specific yellow pills, but you see it's just an allegory. It's not meaning that people with such genes will obtain such color of tablets. It just shows you need to obtain the drugs taking into account your genetic status. And now we know that many of diseases are treated using the data of the personalized medicine, because you keep a lot of money for your insurance company and from your pockets. So how to make the, in case of the cancer, how to make a personalized treatment? Usually it takes 1 month. And in the first day, you need to obtain the histopathological, for histopathological evaluation, the tumor tissue, the FFPE, and something like this. After that, you need to isolate, extract the DNA and RNA, make a library preparation, make sequencing, and after that, you process the data with the specific bioinformatics software. You analyze the targets, you interpret the results, and after that, the specialists, the oncologist, the bioinformatics, the pharmacy, they take into account all the information you obtain from your biopsy, and you obtain from your blood, or directly from the tumor. You obtain the report, the preliminary report, and the final report, which alterations are detected in the tissue of in the biopsy, and you can obtain the treatment recommendation, and so on and so on. So I want also to tell you about the Angelina Jolie effect. She is a famous actress. And also she told to all the world about her problems with health, because her mother died due to the breast cancer, her grandmother died due to breast cancer, her sister has a breast cancer. She analyzed her own genome, and she obtained the information that she has got the increased risk of breast cancer, and in case of the mammary gland, the risk is 27%, and she made the mastectomy in 2013. She removed her breast, and she put there the implants. And after that, in 2015, she made the ovarectomy to decrease the possibility of the ovarian cancer. So it's very good effect. It's very good that Angelina Jolie told us about her breast. So here you see that the 3 to 8% of women have mutations in BRCA1 and 2 genes, and as we know, the 18% of ovarian cancer are caused by the BRCA mutation. So the situation is not so awful, but you know that if you have some relative who died from the breast cancer, you just have to analyze the situation and know that it's okay with you. So, should we hope for Neanderthals? You know, the researcher, the Svante Pääbo, he published the data about the genome region associated with protection against severe COVID-19 inherited from Neanderthals. They have big research. They analyzed the sequences of Neanderthal DNA, and they found that this region of chromosome protects in the COVID-19. And the other work was published with the same researchers, and this work shows that the genetic risk factor for severe COVID-19 is inherited from Neanderthals. We don't know exactly. Maybe it's due to the horizontal transfer of genes, maybe several other reasons. But we know at least that the chromosome 3 is causes the complications of the respiratory system. And this information was very good published in the social networks and on specific sites. So a few words about the genetics in our lives, and we all have or see every day the domestic pets, the dogs. You can see that the dogs were domesticated approximately 30 to 40,000 years ago. And here you see how the cows, the pigs, and so on were domesticated tens of hundreds of years later. So you see the cats, and also we know that all the cats will arise from the Bengal cat, and due to the variation and the geographic radiation, they now live in different conditions. And here you see the different amount of dogs, and you can see it's the ancestral variation, the wolf, and maybe it looks like this species of dogs. Okay, how to obtain the new plant varieties? Long time. And it's not comfortable because you want to obtain the samples as fast as possible. And here you see the picture of banana and the wild type of banana with the big seeds, and you cannot easily eat for sure. The other variety, how to obtain the new varieties, is the mutagenesis, and it's very unpredictable because for sure you can obtain the X-rays, you can obtain specific chemistry, you can obtain the radioactive elements to put them with the seeds somewhere in your laboratory, and you will see how the mutations are generated in the cells. And the other very good method is the transfer of genes from one organism and to another organism. This name is genetic engineering, and I already talked about the Nobel Prize that was elected in the last year. So with genetic engineering, we can transfer the genes from one organism to another organism. Here you see such transgenic plant. They are tobacco plants that live in the laboratory because it's very useful model object. And the other one, the last one, is genome sequencing and it's genome editing. The technology that allows us to make upon changes in the sequences of the DNA of specific species. So here you see the GMO plants that were made in the most of them made in the United States. It's the squash, it's the soybean, it's the corn, it's the cotton, but it's better to say that it's not the cotton, but cotton plants, because the cotton is material. The papaya, and here we see the transgenic papaya, and here are the non-transgenic papaya, non-GMO papaya. The canola that's used for to obtain the canola oil, the alfalfa, the sugar beets. It was invented in 2006. The transgenic potatoes and the transgenic apples. So it's the excuse me, where is my okay. So the GMO plants, what for they are invented, and which genetic modifications are located in these plants. First of all, it's the herbicide tolerance. We will discuss the herbicide on the next slide. The insect resistance, and here you see the cotton that is mostly popular is insect resistant and herbicide tolerant. So you can use the herbicides and these plants will not die. The other two cultures, the sugar beet and the soybeans, are both herbicide tolerant. The corn may be the insect resistant, the herbicide resistant, and the drought resistant. And the papaya is viral resistant, so it was big trouble when the specific virus started to live on papayas, and that's why we're using the method of RNA interference. These genes were neglected. So to the end of 2019, the most of the territory of United States, the most of the samples that we analyzed were made in the most of the territories that was used for the GMO plants is in the United States, and they, as you can see, can produce the seeds of every of these cultivated plants. In case of the United States, the corn, the soybeans, the cotton plants, the rapeseed, the sugar beets, alfalfa, papaya, pumpkin, and potatoes. In other countries, the list is not so long because they just have a small use of the equipment using the GMO plants. So if you tell about the genetically modified soybeans, first we should discuss the Roundup Ready soya. It's it's tolerant to enzyme inhibitor of 5-phosphanol pyrrolylchimera phosphate synthetase. It is very simple molecule, and it inhibits this enzyme that is used in the plants to stop the plant metabolism. And in 2018, more than 22 million of hectares of soybeans grown worldwide. It's very big digit because you can see on the previous slide the most of the territories were in the United States and so on was used to make the to obtain the GMO plants. So this soya is named Roundup Ready soya. So in 2018, more than 22 million hectares of genetically modified soybeans were grown worldwide. So now I want to say that the genetics is definitely a queen of sciences. It can make us healthier. It can provide a food for us. It can make us some smarter. And the biotechnology, the engineering biology, are the parts of genetics that produce results that can be used with the researchers in laboratory everywhere, in medicine, and so on and so on. And I want to show you the profile of Open Doors contest, Open Doors Olympiad. It's a Russian scholarship project, Association of the Global Universities. Our project is named Biology and Biotechnologies. So you can obtain the general biological education and investigate the plants, the animals, and so on and so on, physiology, and you can obtain the biotechnological education. You will investigate the most of the biochemistry, molecular biology, and so on and so on. So the link to the site is on the bottom of the slide. I again want to show you the picture of our beautiful new building of Novosibirsk State University, and invite you to come to the Faculty of Natural Sciences, the Department of Biology, and to Chair of Molecular Biology and Biotechnology. And I was very pleased to talk lecture about this genetics and so on. And if you have any questions, please, you can ask them in the comments, or you can directly go to the website of the Novosibirsk State University and ask questions about the education in Novosibirsk. Thank you very much, and see you in Novosibirsk.
Thank you very much, Dr. Sergey, for this interesting lecture. I'm sure that this information will be helpful for students in choosing their future profession in the university. Thank you very much, and I was very glad to. And now I will tell our future students, some information about Russian scholarship in Arabic language. Thank you very much. يا شباب والصبايا، كان معنا دكتور سيرغي يفغينيوش من جامعة نووسيبيرسك الحكومية. فبدي أذكركم أنتم تستطيعوا تقدموا أوراق على ويب سايت educationrussia.com وتشاركوا في اختيار تنافسي لدراسة في أحسن الجامعات الروسية على حساب حكومة روسية. عندكم تقريبا أسبوعين لتقديم كل أوراق عبر هذه الويب سايت، فتستطيع تختار كل مستويات التعليمية أنت تحب تدرس في روسيا وكل اختصاصات. فتستطيع تختار كمان جامعة نووسيبيرسك كجامعة أولى خلال يعني بين 6 جامعات لازم تختاروا في application تبعكم. فإذا عندكم توجد بعض الأسئلة تستطيعوا تسأل في comments. فهذا كانت بيت روسي في بيروت، ودكتور واديم معكم ضمن نشاطات بيت روسي ضمن علوم وتكنولوجيا في روسيا. أشوفكم قريبا في مشاريعنا الجديدة. شكرا جزيلا على اهتمامكم لموضوع علوم وتعليم آلي في روسيا. نهاركم سعيد، مع السلامة.